Assuming we can make a bacterial chromosome, and we're very far along in this process, it's a piece of inert DNA. What do you do with it? How do you actually boot it up to start a life form from it? With the viral particles, because they're not self-replicating, it's pretty simple. We just put those in, as you saw in the bacteria, the machinery in the bacteria is able to start using that DNA to build viral particles. But it's different when we're dealing with an independent self-replicating cell. You've all heard of nuclear transplantation and cloning, but that's where the entire nucleus in a eukaryotic cell is removed from one cell, and you can put the entire nucleus, all the chromosomes, all the replication machinery, everything in a nice package. That's different than starting with chemically synthesized DNA, where all you have is the naked DNA. So we thought we might have to start with cells where we've destroyed the chromosome either by radiation or some other method, but we decided just to try inserting the chromosome into a cell that already had a chromosome, but by adding certain antibiotic selectable markers, we can then select for what we want. And this is what was recently published in Science. And to us, this is the key paper right now in synthetic genomics because it makes all the steps after this obvious. We started with two species that are relatively close. They're roughly the same distance from each other as man and mouse would be. And we isolated the chromosome from the Mycotes species. We went to great lengths with enzymes that digest proteins to make sure we got down to just naked DNA. And we worked out all kinds of ways to insert that DNA into the recipient cells, the capricolum. And then we just followed this for a while. There were a number of possibilities. One, and we added a marker onto the chromosome as that both chromosomes would be present in the cell, but when you go to, through mitosis, get daughter cells, one chromosome would go with one cell and one the other. And because we have the chromosome we put in is resistant to the antibiotic that we're using, the original chromosome will be killed off, and we'll only have the cells with the new chromosome. Another possibly much more likely possibility is the one chromosome in the cell that we start with has no restriction enzymes. Restriction enzymes are the things, the molecular scissors that chew up DNA. Think of it as sort of the immune system of the bacterial cell. The chromosome we put in actually has a restriction enzyme. We think that restriction enzyme actually gets expressed and then recognizes the parent chromosome and chews it up, leaving just the chromosome that we inserted. We also put a marker in the genome, a set of enzymes that would express a blue so that when it would make it very easy to spot these cells. And here's a close-up of the magnification of this. And it was absolutely stunning. We expected perhaps to get chimeric cells with both chromosomes. We find a lot of evidence for this in nature. For example, when we sequenced the cholera genome, it had two chromosomes that were very different from each other that probably resulted from a cell fusion. But in every case, we only found the transplanted chromosome. And we measured all the proteins. We sequenced the proteins in the cell. We measured all the cell surface markers and found out this was the ultimate in identity theft because all the characteristics of the original cell are completely gone, and the cell turned in just to what was dictated by the new chromosome. So doing this with a synthetic chromosome now, as long as we accurately make the chromosome, it should be no different than what we've just done. And we're in the process of doing those experiments.